Health requires protection. Environmental challenges are part of our lives. We are constantly exposed to a huge magnitude of microbial species that try to use our body as means to survive and replicate. On top of that, we can also face the need to fight cells from our own organism, such as in the case of cancer. The immune system is our tool to recognize anything that's not self. The system is composed of a wide diversity of cell types. Among them, I would like to highlight the T cells. T cells are part of the white blood cells. One particular feature of these cells is that they can specifically recognize virus-infected cells and tumor cells. If you focus on a particular virus, just a small fraction of the T cells we have in our body are specific for it and will recognize it. However, these few cells will respond to this recognition by proliferating, which will lead to the clearance of the infection. Interestingly, some of the siblings coming from this proliferation are able to survive and remain in the body. In the event of a second infection, the cells are then able to efficiently protect us. To be more specific, when a particular cell is able to recognize a virus-infected cell or a cancer cell, it will make many siblings with different destinies. In a very simplified way, these destinies can be of two types. On one hand, we have a large population of short-lived so-called effector cells that are responsible for the effective elimination of virus-infected cells or cancer cells. On the other hand, a smaller population survives long-term and retains stemness, meaning that in the event of a secondary encounter, they can again produce cells with diverse fates. This means that immunity relies on diversity. But how can one cell give rise to other cells with different fates? This is a fundamental question in biology. In immunity, we know that diversity relies on very early events, starting in the initial activation of a virus-specific T cell. Since this encounter, the daughter cells emerging from the first cell division are already different. By different, I mean that these daughter cells have different potential fates. The green cell has a higher chance to become an effector cell, while the blue cell has a higher chance to become a stem cell. Such an event we call asymmetric cell division and can be visualized by fluorescent microscopy. In the video, in blue, we can visualize the chromosomes of the daughter cells. The green structures indicate that this particular cell is still in the process of dividing. And in red, we can easily observe a marker that has its distribution polarized during division. In our study, we have discovered that the extent to which a cell can divide asymmetrically is directly correlated to its stemness, meaning its ability to self-renew, but also be able to generate diverse daughter cells. This means that a cell that can give rise just to a similar progeny is a cell that divides symmetrically, while a cell that can give rise to a diverse progeny is a cell that can divide asymmetrically. We found a way to treat cells such that they can gain asymmetry during cell division. This results in higher asymmetry in cells that could already do that, but even more important, we can store this ability in cells that didn't show it before. While our finding contributes to our understanding of how diversity is generated during T-cell responses, it might also have translational potential. For instance, in adoptive immune cell therapies, in the context of cancer, where tumor-specific T-cells, either inherent or engineered, are in vitro expanded and reinfused into the patient. Our findings would offer the opportunity to imprint diversity into the expanded T-cells, including cells exhibiting stemness, thereby promoting longevity and effectiveness of cancer-reactive T-cells.